afternoon, everybody, and thank you all for coming. It is with great pleasure that I introduce today Vaishali Katju. So Vaishali is an associate professor at University of New Mexico. She did her PhD at Indiana University in Bloomington with Michael Lynch, and she joined soon after the biology department at University of New Mexico. So Vaishali's research interest revolves around understanding the evolutionary fate of gene duplications, of young gene duplicates. So she uses both experimental approaches and bioinformatic tools, as well as C. elegans as a model organism, to look at the extent to which gene duplicates contribute to novel adaptations. I had a chance to meet Vaishali, and to see her at work handling her mountains of C. elegans plates with grace and extreme diligence. And although at that time she was preparing her defense, and we had very little time, that time spent together was very rich, because outside the lab, Vaishali is a very talented artist. She's also a great cook and a good gardener. So, and all these talents blend very well. So today I'm going to present Vaishali the scientist, and Vaishali is going to talk to us about gene duplication in evolution. So Vaishali, you have the floor.
Thank you, it's great to be here. I used to come to Montreal. I think the last time I was here was in 1992 when I was an undergrad in Rochester. I used to just come for away trips in the middle of December for the heck of it, because that's what you do as an undergrad. So it's been about 22 years since I've been here. It's great to be here. I'm glad to see Melania after I've known her for 11 years. It's wonderful to meet the rest of you. I've had a good time. I look forward to meeting more people tomorrow. Can you hear me at the back, or do I need to talk? I'm good, okay. So yeah, my belief in life is to be the Jane of all trades and mistress of none, so here I go. Hopefully I can impress you with that. So I'm going to talk about gene duplication and evolution. I've started doing work a lot more on mutation, not just gene duplication, but that was my first introduction to mutation, was I focused on gene duplication. I'm going to talk about the organization. So my organization of my talk is going to be, first I'm going to talk about some initial bioinformatic analysis I did when the first draft genomes were coming out, just to look at what we saw in terms of gene duplicates. And then some certain work I did on looking at the characteristics of gene duplicates with respect to their genomic features, location, and structure. Then the next three parts of my talk are all based on experimental evolution, because I'm trying to get at more basic, fundamental questions about gene duplication. I'm not getting into the phenotype at all, we're far away from that, but I'm just trying to get a sense of what the rate of gene duplication is, trying to get a sense of what is the average fitness effect of a gene duplicate. Is it good, bad, or neutral? We know that mutations can take an entire spectrum of effects. And then I want to ask one important question as to whether copy number variants, that is both gene duplicates and deletions, are they a common form of genetic change during adaptive evolution? So let's get started. Just some introduction on gene duplicates in evolution. So I start with a really completely speculative story in evolution. I don't know how many of you are familiar with this story. This is called the Snouters or the Rhinogrades. It's a phenomenal book. It came out in the 1950s. It's about, it's a fictional group of so-called 189 species of snouters, or these shrew-like mammals belonging to 14 families, and they live in this remote island in the Pacific of Redpath. So they were discovered by a Swedish explorer trying to escape spies in World War II. And the fascinating feature about this entire group of mammals is that they have an organ, an Auditorium, which is like their nose, and they derive from a shrew-like animal. And they have had this adaptive radiation. So on top you see Optotrix right here, which uses its wings as its ears as wings. It flies backwards and uses its nosorium as a rudder. Another favorite of mine is Orchiodopsis. It uses its petal-like nosorium to attract insects that feed on it. So this trait Nosorium that's evolved in these particular cretaceous mammals serves everything from fighting to feeding to locomotion. And another favorite is right here, the shaggy-faced snouter, which has 4 Nosorium, 2 of which are smaller and thinner, and it uses to uproot roots. But aside from the human elements, and of course the atomic bomb fell on this island and destroyed the Snouters, so that was the end of that. But aside from all the human elements and drama in the story, if you were to ask me as an evolutionary biologist what was one mutation or genetic change that led to this evolution of this novel trait, my best guess would be gene duplication. Let's talk about some real examples of gene duplication. We have many examples. This is just a few. So of course, first is amylase copy number. Increase in the number of amylase copies in dogs since domestication by humans to help them eat spaghetti. Again, a gene duplication in Antarctic fish, this is Trematomus, that they have evolved their entire, from a pancreatic enzyme, a new gene duplicate, a part of the gene duplicated and became a completely new gene that forms the antifreeze glycoprotein that prevents, that mimics the water structure of the water molecule and interferes with it to prevent ice crystals from growing. And this is from a pancreatic enzyme, and it's about 200 to 500 times more efficient than salt. Duck langurs, this is again a group of a subfamily of colobines that live in Asia. While most colobines feed on fruits and vegetables and perhaps even meat in their diet, these have evolved to have a mostly leaf diet, and this has been facilitated by duplication of a pancreatic gene called RNase 1, and this new duplicate copy of that has evolved to allow them greater efficiency in extracting nutrition from leafy materials. Again, a species close to my heart, C. elegans. Again, this is a system, a group of species in the genus that essentially were male-female. They were obligate outcrosses, but in 2 species, and I'll only talk about C. elegans, they have evolved a hermaphroditic lifestyle, where essentially the female, who I consider actually, it's called the hermaphrodite, so they have a hermaphrodite male system, but in effect, I think the hermaphrodite is really a super female, because she's female in every way, anatomically, but has evolved the ability to make her own sperm. And again, that particular gene that leads to sperm production in hermaphrodites is totally different from how sperm is made in the males. And it again arose from, it's called FOC2, and it arose from a partial duplication of a gene of unknown function, grabbed some new non-coding region and made an entirely novel open reading frame. Recently, of course, a lot of people studying copy number variants in humans, and again, a partial duplication of a huge gene family called SR-GAP. One of these duplicates is a partial duplicate of one of the genes, and it seems to inhibit the function of the ancestral gene, allowing neurons to grow further, and this has been implicated in higher brain function in evolution of our species. Again, here, old world primates like us had tricolor vision. This is dichromatic, which is other New World monkeys, though the story is a little more complicated, but most old world monkeys, after they split from New World, about 30 million years ago, evolved trichromatic vision, but extra opsin gene was duplicated and allowed us to see 3 colors, blue, green, and red. Again, a lysozyme that allows, in the bovine gut, that allows more efficient assimilation of their diet of mostly grass. Copper resistance in yeast, again, a gene duplicate implicated in that. Insecticide resistance in Culex pipiens. Again, a lot of examples of gene duplication. So, question is, in evolutionary biology, until recently, point mutations have held sway in being imagined as the most important form of mutation, as being the main facilitators of creating genetic variation. Now, it's not that the role of gene duplication was not appreciated. In fact, Hermann Muller and very early evolutionary biologists like Haldane understood that gene duplication was important. And a cytogeneticist, Susumu Ono, in the 1970s wrote a very important book that kind of resurrected the field. But the problem was they understood the reason why gene duplication theoretically was important, but looking for a gene duplicate is like looking for a needle in a haystack in the pre-genomic era. So after Susumu Ono's book on the evolution by gene duplication, the full impact of gene duplication was not really appreciated until the first genome started coming out. So why did they think gene duplication is important? So, as you know, that there are many constraints on the outcome of evolution. These evolutionary constraints could be physical, developmental, selective, or genetic. Let's focus on the genetic constraints to evolution. Let's consider a parental gene. I'll give a very simple example of a single gene contributing to a single trait. So you've got a parental gene, and the original function is A. Now, it's possible, with further mutations, it could evolve a completely new function, right, and go on to a different evolutionary trajectory. But the problem is, it would always come at the expense of the original function. There are constraints on it. It can't do both very well. So, because of maintaining the ancestral function, this gene is limited in its evolutionary trajectory and prevented in a way. Any mutations that may change its function would be weeded out and it could never develop. But if you duplicate the gene now, you've got two copies, then one can retain the ancestral function and you can have neo-functionalization, the evolution of novel function, because the other copy is free to evolve, take on mutations, perhaps leading to either solutionization, it could be a dead end, but once in a while you get a ruby in the rubbish and you could get a completely novel function B originate. Okay? So that's the whole idea, that the redundancy, that's why gene duplication was perceived as so important, not just by Muller and Haldane, but Joule and Huxley, that it was the redundancy it created that allowed for new evolutionary trajectories to occur. Okay? Now let's extend this to a little more complicated case where we know that many genes have pleiotropic functions, multiple functions. So it's possible that you've got an ancestral gene on the right panel here, it has main function as A, but maybe it has, and many genes have these side functions or moonlighting functions. Let's say on the side it has a small minor activity leading to product B. But it cannot really specialize. Again, the same constraints hold. It cannot specialize and become really good at B, minor function B, without somehow impacting, or in a negative way, the function of ancestral function, the main function A. So here again, gene duplication, similar scenario, the gene can now really specialize on both functions by duplicating, creating an extra copy, and allowing the extra copy to now focus on B function while the ancestor maintains it. Everybody with me so far? You're welcome to interrupt me. I actually like that when people have questions. Please feel free to interrupt me whenever you want. Okay, so let's talk about first. So there was a lull after Susumu Ono's book. We didn't have the tools, the molecular tools, to really get at the full impact of gene duplication till the first genomes started coming out. And I'm going to talk about some analysis I've done with 3 genomes. These are the initial draft genomes that came out in worm, C. elegans, and Saccharomyces cerevisiae, and human. So when the first genome sequences started coming out, the first attempt was to, and this was done by Lynch and Connery in early 2000, what they did was they took the complete set of available putative amino acid sequences. At that time they had 3 full genomes for human, C. elegans, and Melanogaster and Drosophila, and then they had partial genomes for Arabidopsis and the 2 yeast species. So what they did was download each amino acid sequence, do all against all BLAST-P search, had a certain e-value cutoff to identify gene duplicates. Then, what is typically done in evolutionary biology, we prefer not to use sites in the selection to A genes, so we use silent or synonymous sites with third positions often in a codon to actually look at the amount of divergence between 2 homologous sequences. In this case, they're paralogs, 2 gene duplicates. And so that's what Lynch Connery did. They basically used the degree of synonymous divergence within 2 parallels in a duplicate set to get at the synonymous substitution rate. And then, of course, the reason is because it's thought to be largely neutral or invisible for natural selection. And I won't go, there are caveats, there's codon usage bias, etc. But for now, it's a good proxy we have for substitution for the age of a gene duplicate pair. So they were supposed to, the KS or S, as you can see, are thought to accumulate in a clock-like fashion. They're taken as a proxy for evolutionary age. And what they did was they created these maps here, and they found, so if you can see on these graphs, my pointer is not working, on the graphs on top, they have an age distribution. The x-axis is pretty much KS value, or the age of a gene duplicate, and the y is the frequency. And you can see in all the genomes they looked at, you see an L-shaped distribution. A lot of very young duplicates, and as they age, they seem to get lower in frequency. So I went ahead and did the first analysis of, I wanted to know what do these things even look like. We knew nothing about them. So I looked at C. elegans versus part of my dissertation, and then I have since added yeast and and human. Now yeast is a little more complicated because yeast has two kinds of duplicates. Those formed from segmental duplications, but there's also an ancient whole genome duplication event in the species. So the analysis I'm giving you is not from the onologues that were formed from the WGD, whole genome duplication event, but rather just from the segmental. So they're comparable across the three species. We're looking at segmental duplicates, and young, presumably young ones. Yeast, again, there's massive codon usage bias in gene conversion, so they may be slightly older. So what do we see? First, let's look at the genomic location of these guys. So it turns out, of course, we've got very few segmental duplications in yeast, only 21 pairs with less than 10% divergence, synonymous divergence, a lot more in worm and substantial amount in humans. And what we find is, in the case of worms and humans, the majority of them end up on the same chromosome as the ancestral copy. So there's intrachromosomal location of newly arisen gene duplicates. Whereas in yeast, it's the opposite. There is a lot more duplicates found on different chromosomes. Now, question is it because of retrotransposition? Because that could be, you have duplication happening from DNA-mediated versus RNA-mediated events, and if it's from retrotransposition, you expect the duplicate to end up on another copy of the chromosome. So we could do further analysis. In yeast, the problem is that most genes don't have introns, so it's really hard to see one is missing introns or not. But however, there's a significant difference in the genomic location of these young duplicates with low divergence in between these three species, presumably because of yeast being primarily intrachromosomal. The pairs, the two parallels are found in different chromosomes in the genome. What is the extent of duplication span? So in this figure, you can see I have taken the duplication span. I identified for the paralogs, aligned them and found out to what extent did the homology continue. So you have the coding region, but how far along does it go into the flanking regions? And so what was the entire minimum conservative estimate of what was being duplicated? And what we find is that it's kind of similar in yeast and worm. We've got a median, and this is on the log scale because there was no other way I could fit the human data in this. So you can see the human data, the duplication span is huge, which is very surprising to me. Initially, in worms it was only 1.4 kb, and in yeast it was slightly bigger, actually 2.51 kb. Now if you look at the median gene length in the species, you can see that in worms, the duplication span is not meeting the minimum gene length. So you would think that because of this, a lot of duplicates are probably going to be partial duplicates. The duplication span is lesser than the average mean or median gene length. Whereas in yeast and in humans are surprising. So the human study is very recent, we just finished it and we've got it in review right now. I knew that human genes were huge, so I was very surprised. I thought there'd be a lot of partial duplicates, and I was surprised to find out that we had very large duplication spans in yeast, in human about 35 kb, which exceeds the median gene length. And indeed, this will have effects on their structure. But let me wrap up this slide. So it seems there's a high significant difference between species with respect to duplication span, and this is probably mechanistic. There's something about either the repeats that are mediating gene duplication and their spread in the genome will affect as to what gets duplicated. So we have average small-scale duplication events happening in yeast and human, and because they're more likely to. And because the average duplication span is bigger than the gene length in yeast and human, it's more likely to encompass an entire gene. Whereas in worm, it's much smaller. The duplication span median is not exceeding the gene length, so we expect to see a lot more partial duplicates in the worm. So let's see how it actually looks in terms of structure. But first I want to talk about the canonical model for gene duplication. This was actually envisioned by Ono. So what he saw was, here is a gene that has 4 exons and it makes a particular protein out there. And what he envisioned was, when you have a gene duplication, he just assumed, that's what all theoretical models say, there was a complete gene duplication. That means that the entire repertoire of coding sequences and regulatory elements are identical. They're duplicated, leading to the duplicate copy initially making exactly the same protein product as the ancestor. And that's shown here. So now you have 2 copies of the gene and making the exactly same protein. And then the idea is that eventually, when you do have novel functions originating, the idea was that one copy, like I said, will retain the ancestral function, the other copy is free to evolve, gets a whole bunch of mutations, which then impart sequence changes, impart functional change and functional divergence. So this was the traditional model. So the prediction had been, as I began my first analysis of C. elegans duplicates, was that you'll see complete duplicates. And what did I find? I found actually, and this is C. elegans, I found that there was an entire 3 different categories of duplicates out there. Now they were very young. They had, in the region that was homologous between them, that I could align, there was hardly any divergence. Often they had 0 divergence, so they were just born yesterday. And yet, despite that, we had the complete duplicates like Ono suggested on top, where here you see a gene with 3 exons, the entire, at least the coding region got duplicated. But we also had partial duplicates where one copy had unique sequence in red to the exclusion of the other copy. Okay? Presumed, and we of course can't always tell which one is the ancestor, so that's an issue. You have to have some kind of ortholog in another species to tell the direction of duplication. But we call these partial structure, and then you had instances where there were chimerics. Both copies had unique exons and or introns to the exclusion of the other copy. So right away, even with very low synonymous divergence, in the part that is homologous, the yellow part, that's shared between them, they could have absolutely no sequence divergence in those areas, and yet in other, they could have completely new exonic regions in some cases, in the case of partial and chimerics. So, and we were shocked when we looked at worm, we found that the partial and chimerics in the middle, actually 60% of an entire set of 290 young duplicates in C. elegans. So we were shocked that only about 50% or 40% were complete duplicates in worms. So this is right away an immediate way to give new function. You've got entirely, at least 60% of the new duplicates have completely different coding regions, or at least parts of the gene open reading frame have completely new exons. Now it's very different since we've done this analysis in yeast and human. Yeast, they tend to be mostly completes, right? And in humans, again, we found, we were surprised. Again, a lot of complete duplicates, a few partial and chimerics. So it's totally different. Again, when we look at the different species, more than 80% of yeast and human paradox are complete, have complete structure, structural resemblance. But in worms, it's highly contrasting. About 60% of them are structurally heterogeneous. They either are partial or chimeric duplicates. And again, we found a highly significant difference between species in the frequencies of these 3 categories. And now that makes sense with the duplication span. Remember, for yeast and human, the span exceeded, the median span exceeded the average median gene length, but not in worms. And so you can see in worms, you see a lot fewer complete duplicates, a lot of partials and chimerics being formed. And then finally the conclusions. I'm not going to present much more, but there were contrasting patterns of genomic location between the 3 species we've looked at. In yeast, they tend to be mostly on different chromosomes when they are presumably young, and in human and worm, they tend to be intrachromosomal. Again, we know that a lot of the yeast duplicates occurred via polyclonization, early whole genome duplication event, and we expect the copies to be on different chromosomes if that happens, but we've excluded that. We only looked at small segmental duplications, and even those are on different chromosomes. But what about the yeast paralogs from small scale events? So we don't know why this is happening. The mechanism is different. We know from experimental evolution studies, MA studies of yeast duplicates done by Lynch, that in fact it tends to be, even small scale segmental duplication events tend to be frequently translocative. They end up on different chromosomes right away from experimental evolution studies. So there's definitely different mechanisms of duplication in worm, human relative to yeast. In terms of structural resemblance, again, very contrasting patterns of structural resemblance between paradox. An average small-scale duplication event tends to, in yeast and humans, yields often an entire open reading frame, leading to a complete duplicate, but not in worms. Worms, you see a lot more partial and chimeric duplicates. Now, of course, there's always the issue of role of natural selection in these guys. Remember, these 3 genomes, these are draft genomes. These are presumably lab strains or wild isolates that have been under selection. So you have no idea as to how many, the duplicates you see are what has survived. You have no idea as to what may have arisen, has been lost. For example, the fact that we only found 21 pairs in yeast of small scale segment, does that mean that yeast has a very low duplication rate? Actually, it's not true. Yeast has a huge duplication rate, but because the large effective population size, almost reaching to the levels of bacteria, is 10 to the 7 in yeast, the effective population size selection is very efficient. We guess that they are being weeded out. There's a very high duplication rate, but in draft genomes, so genomes under selection, you don't see them. They're being weeded out. So that was our first hint that something funky is going on. Whereas in worm and humans, you can see the effective population, the long-term human effective population size is only about 10,000 individuals. So worm and human will not have as good efficiency of selection to get rid of duplicates if they're slightly deleterious when they arise. So we probably see a lot more worm-human duplicates surviving than we see in yeast. So I'll wrap up. That was just the first draft from the bioinformatic analyses. Now I'm going to get into experiments we've done. So first is, what was the actual rate of gene duplication? How important is it? Is it surpassed by mutation rates? Is it the main contributor to genetic variation? So we tried to get at that. And for all these three, yeah?
Question before you get into the experimental stuff. The L-shaped curve of the age distribution, many of the parallel, the duplicates are young.
Yes.
And at first I thought, oh yeah, that's because, well, most duplicates are probably going to be weeded out because you're not going to get a lucky mutation that's beneficial. But then I realized, no, these are fixed.
Yeah.
Okay,
so
how can you explain?
So a very high duplication rate. So even though a lot of them are being weeded out in yeast because the effective population sizes, so it's very efficient at weeding out even very small selection, negative selection coefficients. In worm and yeast, they probably don't get weeded out. In worm and human, they don't get weeded out as fast because we have smaller effective population sizes and our selection is not very efficient. But despite that, even though you may lose a bunch at the beginning, the rate is so high, and that's what I'll get at.
But it's interesting, though, I think you're sort of saying that these duplicates that will eventually be eliminated first can be fixed, and then later eliminated, which to me is a little contrary to...
No, but remember, this was a genome, one individual. Often the first draft genomes came out. We don't know if they're fixed in the species.
Oh, you're not sure.
Right.
Okay,
This was draft genomes. This was the first C. elegans genome, probably, presumably one individual. Same with these humans, it was probably Craig Venter's genome.
But that would be an interesting study to know that, because I
Right.
think that would be...
Right, and that's again, that's a huge, the next step is to see so many die out, as which ones survive and which ones, what influences their fate, and are there certain determinants that influence their fate. That's a future study, absolutely. So let's get at the first empirical estimate of the gene duplication rate. And we used, so the next three studies are entirely experimental. We use worms because they're so tractable, they have a very short generation time. So why do we care about the gene duplication rate? Because we care about mutation rates, and the gene duplication rate creates natural variance in gene copy number. Okay? And the rate of fixation of copy number variance, like any other mutation, depends not only on the input due to mutational input due to the mutation rate, their spontaneous rate of origin, but also the probability of fixation or loss versus natural selection or genetic drift. So the idea we are proposing is that if there was selection for increased gene dosage, this is the case with gene duplication, if you want more dosage, more copies of a gene, if there's selection for that, and if the duplication rate is greater than the point mutation rate, then your template, what you have, the material you have is a lot more duplicates, so gene duplications are likely to be fixed first. If the duplication rate surpasses, there's a lot more genetic variance created by gene duplication. And if there's selection for increased gene dosage, you will likely solve a problem by fixing the duplication first. Okay? So let's look at, this is not a new idea. Empirical estimates of the duplication rate have been going on for years. John Roth, who was a bacterial geneticist, was doing this as early as the 70s. But because they didn't have the tools, often they were locus-specific rates. So we have locus-specific rates, empirical estimates. We have them in bacteria, Drosophila, and humans. They're very high, ranging from 10 to the minus 3 to 10 minus 7 per gene per generation. These are huge. Mutation rates are often low because they've evolved to be low, unless you're a virus. But even then, so the average point mutation rate is about 10 to the minus 9 per site per generation. So these were orders of magnitude higher than point mutation rates, what we know of them as. But the problem was, from these estimates, is they're specific to certain, and there's often a bias. You pick loci that may have fast rates. So there's always a bias as to whether these rates can be generalized to the entire genome, all loci in the genome. Okay? Of course, then you had another rate coming out. This was the first bioinformatically genome-wide derived rate. And the way this was done was in the Lynch and Convery paper. They had done the bioinformatics study of these genomes, those 6 genomes I showed you, which showed the L distribution of duplicates. And the way they did it was very similar to life history tables in ecology. They looked at the youngest duplicates, about 10% or less, and from that they looked at their frequencies. They divide them to age cohorts as such, like you do those life history tables. And they looked at different cohorts and tried to figure out what the rate was. Now the problem with this kind of this is what they did. So those were the 6 genomes they looked at, and this is the rates they came up at by looking at gene duplicates with less than 10% divergence. They estimated C. elegans they're quite similar, 10 to the minus 8 per gene per year. We know the point mutation rate in C. elegans, so we can convert that to 10 minus 9 per gene per generation. But roughly 10 to the minus 8 per gene per year. Now when these estimates first came out, it was a hue and cry, because now they were pretty much the same order of magnitude as point mutations at the time. And it's a bit of an apple and oranges comparison. Point mutations are per site per generation, these are per gene, but there's no other way to compare them. But the fact that there was so much variance being created, 10 to the minus 8 per gene per year, was huge. So at that time, Sally Otto said this is an astronomical rate of gene duplication. There was gasps, and yet I could argue that their selection, all these genomes were not controlled lines. They were either lab strains or wild isolates. There's selection acting in them. This could be an underestimate, right? Because there could be a whole slew, a whole population of gene duplicates that have been weeded out before you even see them, okay? So the best way to do this really is to get the direct rate, and that's from mutation accumulation experiments. And just these experiments, and I think I have a slide to show what the whole idea, or what the theoretical, why we use MA experiments, and I know that Melania uses them quite a bit with the Daphnia lines. So the whole idea behind these experiments is you just maintain lines under repeated bottlenecks. At the minimum you can go. And by doing that, you remove the power of natural selection, and to remove any deleterious duplicates, and you pretty much let the organism be a vessel for just collecting duplicates, for collecting mutations. Now, of course, yeah?
I just have a question about that rate. So that's an average rate, but I've just been thinking about like whole genome duplication, right? So that could be an extremely rare event.
We're talking about segmental, right? So I'm not talking about polyploidization events. And they are rare in also animals that have differentiated sex chromosomes, because it creates
instantaneously duplicates every gene.
Right. So I'm talking about just anything that is not a polyploidization, segmental duplications, and they can range from part of the gene all the way to you can go up to aneuploiding an entire chromosome. Right. So the whole idea behind these experiments is that you remove the power of natural selection and you essentially drift rules. Genetic drift is the main evolutionary force, but essentially your organism, your individual line, is a vessel